which is that so that's probably storing the optical actors. Chair, the examination chair.
I'm happy that they're starting. You were. Okay, sorry.
Good morning. Okay, bagel people, 2 minutes warning.
And then talk about what?
No way.
Dr. Brown is ready, which means everybody is ready now.
Yeah.
No?
Down first, and then I'll be.
Sit and
Okay,
down.
now I'm ready. Thank you.
Thank you very much. All right, good morning, everyone, and good morning, people on Zoom. I would like to welcome you to our Friday seminar. Again, it is time to celebrate not only holidays, but also a vision research. And today we have fantastic visitor from far, far away. And with that, I wish you a wonderful holiday season. We have one more seminar a week from now, and I hope we all will join and then celebrate the rest of the year. I hope it was very successful for everyone. All right, with that, let Deepak introduce our speaker, please.
Good morning and welcome to the Distinguished Speaker Seminar Series of Kevin Herbert Ice Institute at UC Irvine. My name is Deepak Phoria and I'm an assistant project scientist in Vladimir Kapelostyan. It is my great honor to introduce our today's speaker, Dr. Phyllis R. Robinson. Dr. Robinson is a professor of biological sciences at the University of Maryland, Baltimore County. She began her science journey by earning a BA in biology from Wellesley College, Massachusetts in 1973, and a PhD in zoology under the guidance of Dr. Derek Bowens from University of Wisconsin-Madison in 1981. She then completed her postdoctoral training in biology and biochemistry in the lab of Dr. Daniel O'Brien from Brandeis University, Massachusetts in 1992. And she has been a faculty at the University of Maryland, Baltimore County since 1992. Dr. Robinson has earned many prestigious awards and honors during her career. An NSF pre-doctoral traineeship in 1974 while she was at University of Wisconsin-Madison, NIH Research Service Award Fellowship in 1983 while she was at Brandeis University, Mary Bunting Institute Science Fellowship in 1990, University System of Maryland Board of Regents Faculty Award for Excellence in Mentoring in 2000. Leadership Alliance Mentoring Award in 2006, UMBC's President's Commission for Women Achievement Award in 2012, Marilyn E. Demarest Award for Faculty Advancement in 2018, just to name a few. Dr. Robinson's research has contributed immensely to our understanding of the visual pigments. Prior to the discovery of the crystal structure of the inactive and active form of rhodopsin, using the site-directed mutagenesis, her group was the first to identify the role of lysine in the seventh helix and glutamic acid at position 113 in rhodopsin activation and deactivation. In 1992, she published the first ever description of a constitutively active G protein coupled receptor. Her studies of the melanopsin established that this pigment belonged to the G protein class of receptors, revealed its spectral properties and photochemistry, and the deactivation mechanisms. Dr. Robinson also has a long-standing interest in the evolutionary adaptation and spectral tuning of the visual pigments. She was the first to show that dolphins and seals are monochromats and that aquatic mammals tune their visual pigments depending upon their foraging depth. So without further ado, let's give a very warm welcome to Dr. Robinson.
So this is so thank you for the invitation. It is great to be here. Thank Vladimir, Chris, and Greshna, especially Greshna sent me a wonderful email saying, come get out of the cold and come to where it's nice and warm. So it's a pleasure to be here. And it's being recorded, I got it. So what I hope to do today is introduce you to the molecule that we've been working on for the last bunch of years, and share with you 3 stories. One part of published, and the two others are sort of in press or being worked on. So let's see. Do you not want to move? So what I'm going to do today is give you a brief introduction to melanopsin and intrinsically photosensitive retinal ganglion cells, and then talk about 3 stories about the regulation of mouse melanopsin phototransduction and its crucial role in non-image forming from molecules to behavior. So as I probably don't have to impress with you, that vision is really important, and probably a lot of you deal with vision and tetrapods, but the ability to detect light is really an important sense that species have evolved lots of different mechanisms for light detection. And so clearly it's a sense that's really important. And sometimes we can learn stuff from interesting things by looking at different species who have solved the problem of phototransduction in different ways. So there's actually 2 kinds of vision. There's image forming vision, which we're all very conscious of and aware of, and that we are using today. But then there's non-image forming vision. And it is involved in things like circadian rhythm. So you all probably got up at 6, 7 o'clock. I was up at 4 o'clock this morning because that's 7 o'clock my time. And so it would take me a week to photo entrain to your light dark cycle. It's also involved in the pupillary light response, probably seasonal affective disorder. You probably don't suffer from it, but if you went to Norway, you might. Light effect on mood, a whole host of things. So as we know, visual pigments are a classical G protein coupled receptor, but they're perfectly designed protein switches. What's unique about visual pigments among all G protein coupled receptor is 11-cis-retinal, which is an antagonist, is covalently linked to the GPCR. And the light in 200 femtoseconds creates an agonist. And that's the only direct effect of light in all of vision, which I still find totally amazing. And so it's this, and the visual pigment that Chris first got a structure of is so tightly bound with the antagonist. It signals thermally once every 125 years. So it's a really beautiful switch. So as I said, G protein, opsins are part of G protein coupled receptors. And we can learn a lot about GPCRs just by studying visual pigments. So the molecule I want to talk about today is melanopsin. This is a homology model we made based on the squid opsin. Just to point out that melanopsin is more related to the visual pigments in the fly or a lobster than it is to the ciliary opsins we have in our rods and cones. And so we have to think differently than rod or cone phototransduction. So the melanopsin is expressed in a small subset of ganglion cells. And what's also interesting, and for students, don't ever take that a field is mature. People have been studying the retina for 150 years, and it was only 24 years ago that Ignacio Provencio found melanopsin in a frog skin and then in a frog retina at Yusuf outside of Washington. So there's always things to learn. So, it's amazing. The IPRGCs, or these ganglion cells, are really interesting. One, they don't look like rods and cones, which have elaborated anatomy to concentrate the visual pigment. So like rods, 90% of the outer segment is rhodopsin. This is a regular ganglion cell that expresses a visual pigment, but it also integrates information from rods and cones. And so it's a tiny computer that has its own phototransduction cascade. These cells, even though there are few of them, they tile the retina. This is a picture of a mouse retina with melanopsin antibody. These cells, in the mouse, there are 6 anatomical subtypes, and they project to all sorts of areas of the brain. And so light information is being, non-image forming information is being transmitted to many nuclei in the brain. It also turns out that these IPRGCs are involved in visual image forming vision and probably involved in contrast. So just to point out the difference between IPRGCs and rods and cones, you can see that rods and cones hyperpolarize rapidly and respond to light, whereas these IPRGCs depolarize and they fire action potentials. And so, and their kinetics are really very different than rods and cones. And what we're going to see is that rods and cones are pretty homogeneous. They may have different G protein coupled receptor, different visual pigments, but the cascade in rods and the cascades in cones is pretty homogeneous. That is not the case in IPRGCs. And so because it's an R opsin, initially the canonical phototransduction cascade that people thought about was a GQ phospholipase C TRPC7 pathway. But it turns out as we do more, people have done more physiology, there seems to be a real heterogeneity in the phototransduction cascade. And this is a controversy that's hasn't been solved. And then my latest hypothesis is that there are 2 phototransduction cascades possible in all of these 6 subtypes, and some are maybe emphasized in one subtype or other. The deactivation cascade is very similar to G protein coupled receptors, where there's a kinase that phosphorylates the tail, and arrestin binds and inhibits the signal. And it turns out that it's not visual arrestin. It's either beta 1 or 2, which means that there are clathrin binding sites, and it's highly likely that the melanopsin may be endocytosed, and we don't know if it signals as it is endocytosed. So what I want to do today is give you 3 stories about the regulation of melanopsin phototransduction. The first, we've published a lot. I'm going to give you some more recent elaboration of that, and then talk about pKa phosphorylation, and then the role of RGS proteins. So to begin with, I'd like to talk about melanopsin and phosphorylation and deactivation. So mouse melanopsin is really very different than rhodopsin. It has got an extensive tail. It's 171 amino acids. It has 38 serines and threonines that are potential phosphorylation sites. So we did early on an experiment where we mutated all these serines and threonines to alanine and then used a heterologous expression system and measured calcium. And we can see here that the kinetics of the response in red are very slow compared to the wild type melanopsin. And so we published a series of paper. One we collaborated with Chris on, looking at the dynamics and the mechanism of the biochemistry. What I want to show you today is how this reaction affects both the physiology and the behavior of a mouse. And so what we did was we took a mouse that has Cre recombinase behind the melanopsin promoter, and then we injected a AAV virus that's Cre dependent, and so it's only going to be expressed in the iPRGCs. And here you can see that the virus had a mRuby to show us that we actually infected cells. And you can see that with the yellow that many of the cells that are infected are also expressing melanopsin. And so what we did is we took this was done with my collaborator, Lane Brown at Washington State University. And we did whole cell recordings, and you can see the wild type in response to a flash. We fire action potentials, but then it turns off fairly rapidly. Whereas the mouse with the mutant melanopsin that has no phosphorylation sites, it fires action potentials for about 15 minutes. And then what we did is we used a multi-electrode array and confirmed this with a whole series of ganglion cells. And you can see the difference at the top is the wild type retina, and then the bottom is the mouse with the phosphonull mutant. So then we wanted to take a look at a behavior, and the behavior we were looking at was the pupillary light response. And to just remind, what did I do? What happened here?
We have someone else sharing. Is them here?
I think that starts. Let me try again. Sharing? I think you just need to reshare the screen over here. Just sharing from here, is that right?
Are you still on the Zoom, Phyllis?
She's sharing through the systems at the room Zoom.
Or just share the screen with the-
What?
Then we do this.
Someone's sabotaged.
Oh, wait, I guess we're. Okay, so the pathway that we used was a pupillary light response. And to remind you that light finds its way to the olivary pretectum and then back to the iris on both sides of the retina so that we can use a consensual pupillary light reflex assay. So we can shine a light on the one eye and then record with a video the pupil dilation, constriction and dilation. And so the way we did this experiment, we used our mouse as our control. So all mice were Melanopsin knockout. We assayed them for their pupillary light response. You can see that here. When you shine a light, then rapidly after the light's off, the pupil dilates. And then what we did was we injected both the wild type virus or the Melanopsin mutant mod, and then assayed the pupil light response. And what you can see here, this is the kind of results that grad students really liked because it's so significant. With the wild type compared to the mutant stays constricted over here over 90 seconds. And then when we did the experiment, it turns out that it is 90 times slower to dilate. So it took 45 minutes for this mouse to dilate its iris. I don't have time to show you today, but the mice also photoentrained more rapidly. So it turns out that not turning off Melanopsin has significant results. And so we showed that phosphorylation controls its kinetic and it has effects on both the physiology and the behavior. So then we were curious, these were mutants that we made in our laboratory. What about nature? Has nature modified the C-tail of Melanopsin in animals with different architecture? And so first thing we found looking at the literature is depending on the type of rod cone architecture, the fewer cones, the more IPRGCs that we find in the retina. And then when we aligned the tail, we looked at the serines and threonines, and we found that as the animal lost some of its cones, there are fewer serines and threonines. And what was really interesting in an armadillo, site 382 is a proline instead of a tyrosine. And so we've assayed in a heterologous expression system all those Melanopsin, but today I just want to show you the armadillo. The armadillo is a rod-only mammal. And it turns out, you can see in dark yellowish green, its native Melanopsin is slow to activate and very slow to turn off. And so this is an animal, if it got into the light, it probably would want its pupil constricted because it has only rods, it has no cones. And so it seems that the altering the serines and threonines on the carboxy tail is something that nature evolution has worked on to control the kinetics of this molecule. So a second phosphorylation that we were interested in studying was pKa. So when we looked at the sequence of Melanopsin, there are 3 canonical pKa sites in the loops. And so it appeared, we were curious to know, was there a cyclic AMP pKa dependent reaction? And so this is work that is about 10 years old now. But Joe, we added, this is a calcium imaging assay. We added just 8-bromo cyclic AMP, which is a non-hydrolyzable cyclic AMP analog. And you can see that the response has been muted, right? So it's not as robust as the wild type Melanopsin. We've gone on to characterize that, but what I want to talk about today is how we think that reaction might be involved in controlling some of the physiology of IPRGCs. So work from Michael Doe, who I think talked here earlier, he found that there's a population of IPRGCs that signal in response to different timeframes and intensity of light. And so it's an ensemble effect, and it's not just one IPRGCs that's giving the superchiasmic nucleus the information about light. And so we were curious to figure out the mechanism behind this. So we have, when we looked at the IPRGCs, we see that several of them, M1s, express the dopamine receptor, where there are others that don't express D1. This is the D1 dopamine receptor that triggers a stimulation of adenylate cyclase. And what we found is, what we hypothesize is that in the cells that were able to signal over a long light intensities, that they were being, melanopsin was being phosphorylated, so the extent of its signal was depressed, and these cells didn't go into early depolarization block. So basically, by phosphorylating melanopsin, you get less of a light response and you don't go into depolarization block as early. And so when we did this experiment, we were recording from a retina, and you can see in the absence of dopamine, the cell goes into depolarization block. In the presence of dopamine, it fires through the whole time. And then if we put on a dopamine D1 antagonist, we inhibit the response. And you can see this in single whole cell recording. In the presence of dopamine, we get a smaller light response than when we put on a dopamine antagonist. And it turns out that melanopsin response is also modulated by a GS dread. So this is a AAV dread virus that is Cre-dependent, injected into mice, melanopsin heterozygous with Cre. And when we record the light response and then we put in CNO and stimulate the GS dread, we see a diminished light response. So this is sort of consistent with our hypothesis that PKA, or cyclic AMP dependent process, is modulating melanopsin signaling. So then what we wanted to do is look at the behavior. And so in this experiment, we have mice that have the PKA sites mutated, and we're looking at photoentrainment at high light intensities. And what you can see in this mouse here, that in response to 1,000 lux, it doesn't photoentrain, it just free runs. And so what that suggests is that because there's no PKA phosphorylation, all these cells are in depolarization block, and the IPRGCs are not signaling to the SCN to photoentrain. So we conclude from this story that melanopsin is phosphorylated by PKA, which is activated by cyclic AMP. And this phosphorylation may be part of the reaction allowing M1 IPRGCs to function over a large range of light intensities. So the last story I'd like to talk about today is the role of RGS proteins on melanopsin signaling. So it turns out that if you look at the electrophysiology of IPRGCs, that there's a heterogeneous response. And then like M1s that express a lot of melanopsin have a really robust response. And it's very different with M2s and M4s. The kinetics are different and the size of the response is different. And so unlike rods, which if typically you'd have a population of rods, there's a very stereotypical response to a light response, the same with cones. Here, there's a huge heterogeneity. So we were curious, how can melanopsin signal such heterogeneity in vivo? So they're differently expressed phototransduction cascade components, different G proteins. different regulators of G protein, different channels. How can a single GPCR elicit different signaling outcomes? That's the question we were asking. So my grad student, Lee Harkless, decided to focus on our RGS proteins. So we know RGS proteins are in rods, or the rate limiting reaction that controls the kinetics of the light response. So this seemed like a reasonable target for us to look at. And just to remind you, RGS proteins are gap proteins. They speed up the endogenous GTPase activities of G proteins. So G alpha G proteins hydrolyze GTP, but it's a very slow reaction. And so RGS proteins help catalyze that. And you can see that in B up there, where this is a muscarinic receptor activating GO in the presence and absence of a gap protein. You can see the kinetics are really affected by RGS8. So what we did was we took a look at the, there's RNA-seq data that was published by Tran, available at the Broad Institute, and we analyzed it and looked at melanopsin expression and RGS expression as a function of these different subtypes. And what you see is that this RGS16 seems very enriched in M1s, and it's not highly expressed in any of the other IPRGCs. And then when we did some RNA-seq, RNA scope, in addition to using melanopsin antibodies, we quantified the expression of this RGS16, and it's highly enriched in one population of M1s as opposed to the other IPRGCs. And then we looked at differential expression of RGS5, and RGS5 is also highly expressed in a different subtype, M4, and it's not very highly enriched in all the other subtypes. And so we have, we did the same experiment, and this one, we're looking at RGS5 RNA scope with melanopsin antibodies. We quantitated, and it turns out that most of the antibody RNA the RNA for RGS5 is predominantly in M4s, and so it's highly enriched in M4s. And so if we look at also the G proteins, what we find that all G proteins except G15 and GNL1 are expressed in all the IPRGCs, but there's some heterogeneity in the expression of the RGS proteins. So M1s have RGS4 and 17, which are ubiquitous to all the IPRGCs, but M1 is enriched in 16, and M4 is enriched in RGS5. So then what we wanted to do is look at the melanopsin signaling profile. And what we did is we used a Brett assay, and we used some HEK cells where all the G proteins except GI have been CRISPR'd out. And then if we use a pertussis toxin, we can control which G protein is being expressed in our HEK cells. And then what we can do is we can look at the Brett assay under a variety of conditions, and we can assay every G protein that melanopsin can activate, and then look at the effect of RGS proteins on this Brett assay. And so here is a radar plot. These are plotting the highest Brett signal. And what you can see is that melanopsin is promiscuous. It can activate many G proteins. It activates the GQ family better. And you'll note that it can activate G15, but G15 is not expressed in these. Activate GI and GS. And so then what we did is we assayed these melanopsin in the presence of different RGSs. And so here we have RGS 16, which is enriched in the M1 population. And we see that in the presence of RGS 16, 50% of GQs are inhibited, but only 30% of GI, which is an inhibitory G protein for adenylate cyclase. When we looked at G5, RGS5, we see that its effect is predominantly on GIO, although it doesn't inhibit GQ. And then when we look at the RGS17 and RGS4, they regulate broadly, especially GQ. So what we decided to do is create in our heterologous expression system an artificial M1 or M4. The idea that it's these RGS proteins that are going to give us different G protein activation. And so in the dark, the black, that's melanopsin without any RGS proteins. And what you can see is that if we recapitulate an M1 or M4, that we get a differential expression of either GI or GQ. And so in the M1 condition, where the RGS are predominantly inhibiting the GQ G protein, whereas with the M4, RGS 5, 17, and 4, they're acting on GIO. And so what we can see is that we could imagine that we could modulate the signaling of the same GPCR by differential expression of a RGS protein, which would bias the signal. So it's an ability to bias the signal. And so we were interested in why M4s might want to regulate the GI with the inhibitory G protein, which is involved in regulating cyclic AMP levels. And so there were in the literature, there was a suggestion that in IPRGCs, that's the beta gamma of GQ that is can activate adenylate cyclase 2. And so we did an experiment in our HEK delta 3 knockout that don't have any G proteins in the presence of pertussis toxin. And then we add GQ and we measure cyclic AMP using a glow sensor assay. And what you see is as we transfect more GQ, we get a increased fold in cyclic AMP, which we can inhibit with a protein that scaffolds G beta gamma. And so what you can see is that it is possible to stimulate cyclic AMP just with GQ beta gamma in our heterologous expression system. So then we were curious, can we stimulate the alter cyclic AMP level and look at the physiology? And so what you see here is that we're using dreads that are expressed based on either Cree or Flox dependent, so we can use intersectionality to target the IPRGC we're interested in. And so if we use a GS dread in the dark, both M1s and M4s fire action potentials, and the firing of action potentials in M1s is really unexpected because several papers have suggested that there's no cyclic AMP biochemistry going on in M1s, but clearly this cyclic AMP is able to activate some channel or some cascade, and the same in M4s. Both M1s and the kinetics are a little different, but you see that we can GQ will elicit action potentials in both M4s and M1s and also GS. So we found that really interesting. And then we did an experiment where heading back, this is the one I showed you before, that if we stimulate cyclic AMP levels with GS, again, we affect the light response. We're thinking this is through a pKa dependent process. So to conclude this biochemistry, we see that, let's see, where's my, oh, I missed it. Okay, there's a slide missing. When we did this experiment in the looking at photo entrainment, it turns out that yes, so we haven't modulated the behavior yet using the RGS proteins, and that's where we're attempting to go. So our conclusions from this experiment are that RGS proteins are differentially expressed in M1 and M4 populations, and they can regulate melanopsin signaling. The enhanced deactivation by GIO by RGS in M4s may be conducive to cyclic AMP generation, and that melanopsin GQ beta gamma can increase cyclic AMP through A2, and that GS dreads drive action potentials in both M1s and M4, and increased cyclic AMP in M1 may result in the adaptation mechanism that reduces the photocurrent. So our overall conclusion from all these three stories is that both light-dependent and cyclic AMP-dependent phosphorylation affects melanopsin signaling. These modifications impact both the physiology and the behavior. And we're very excited now about the role of RGS proteins, and they may convey a system bias. And so many of the properties of photoreceptors are mediated by the visual pigment, its regulation, and the regulation of the phototransduction cascade that they trigger downstream. So I'd like to thank the Robinson Lab. Besides science, we do have some fun. And just to show that sometimes PIs get into the lab, is Samar, Hattar, and Tudor doing some, preparing some mice for a pupillary light response. And here are the funding agency, NSF and NIH, and the grad students that I've worked with. And there's been 62 undergrads that have been in the lab. And my collaborators, Samar Hattar and Lane Brown, and other people. And I'm going to end with a shameless advertisement for a meeting that I'm organizing in June outside of Boston. The Honorable Christoph Polcheski will be there, so please come and maybe you can see him in the basketball court.
Thank you, Phyllis. Let me start with two very quick questions. One is, did you look particularly in humans? Do you know if melanopsin in those different M1, M2, and 4, the level is regulated by light, whether you have expression level changes?
Oh, that I don't know. That I don't know. In mice, there doesn't seem to be any indication that darker light rearing affects its.
And in humans, do you remember the number of cells that are present, M1, M2, M4?
Well, so in humans, there looks like there aren't 6 subtypes and maybe 3 subtypes. And I think there is a differential expression in those.
Mm-hmm. And how many? How many cells really regulate our mood and pupillary responses?
We're probably talking somewhere between 500 and 1,000 cells.
Is it not amazing?
Great talk, Phyllis. Thank you. So my question is, once melanopsin signals, is there any internalization that happens? Is there any signaling through arrestins
So
that
that's a
bind?
really great question. We've shown that HEK cells, we can see internalization. The problem with, our heterologous expression system, it's great for easy experiments, but the ratio of proteins to regulatory is not what it is in the IPRGCs. And so we're trying to figure out some experiments to actually look at internalization in the actual IPRGCs.
Yeah. Okay, thank you.
But yeah, I think it's a real possibility. And then with so many serines and threonines, the whole idea of bias signaling with arrestins seems like there's a huge potential. And the other big question is, what's the rest of the tail? We know that we can cut off a third of the tail and not affect the phosphorylation kinetics. And so what's the rest of the tail doing? I don't know. We're trying to do some pull downs to see what other proteins might be binding to it.
So in photoreceptors, when RGS9 deactivates phototransduction, this deactivation also depends on the effector protein that is PD6, right? So do you see any difference in expression of the effector proteins in M1 versus M4 that would affect the kinetics of the activation?
So there's a different RGS that we've looked at. We don't, what we see, there is a difference in G protein expression. We haven't looked at some of the other adaptive proteins.
Hi, my name is Hewitt. I'm wondering if there's variation in the kinases as well that between or across the different ganglion cells. And could that potentially have an effect?
So there's, there's 7 families of G protein coupled receptor kinases. So my hunch is that it's not, melanopsin doesn't have its own kinase like rhodopsin. There are other GPCRs that are expressed in these ganglion cells. And in fact, an experiment was done by Pander. If you infect all ganglion cells with melanopsin, they all can become light sensitive.
In terms of the phototransduction kinetics, could variation in the expression of GSKs affect the
Yeah, I mean, we don't know what is the right limiting step. I mean, we could, do what was done with rods and overexpress RGS proteins and overexpress the kinase. In rod system, it turns out it's the G protein is the rate limiting step, not the phosphorylation.
Hi, Phyllis, beautiful talk. You show us these images of RGCs having melanopsin spread throughout the cell, in the cell body and also in the dendrites, right? And you have this long tail in this molecule. Is it possible to make experiments showing if there is any difference in the activity of this melanopsin in the dendrites versus the cell body?
So, that's a good question. I don't know. I mean, Michael Doe's looked at the transduction cascade through comparing the soma and the dendrites, and I don't think there's huge difference in the kinetics, but if I remember, I'd have to look that up.
Hi, Dr. Robinson. Thank you so much for your wonderful talk. So my question is about the IPRGCs and the pupillary light reflex, because it has been found that the cones and rods contribute to that reflex, especially to the early and the dynamics of it. So I'm curious, especially because you have RPE65 knockout mice and patients with LCA that have impaired reflex. So how do you perceive these different cellular contributions?
Well, okay, so in terms of to get a prolonged pupillary light response, you need melanopsin. And one thing we don't know in terms of RPE65 knockout is what's the visual cycle for melanopsin. So melanopsin is ganglion cells very far from the RPE? Is it Mueller cells, or is it retinal, the retinoids diffusing down to the ganglion cell layer? I don't think that's been definitively answered. And it turns out that there's a recent paper by Corinne Baer from Samar Hattau's lab. It's rods that predominantly feed into the pupillary light response.
Thank you for that really interesting talk. So going back to kind of the heterogeneity of IPRGC responses, I was kind of curious if anyone's seen patterns in like IPRGC responses on a single cell level related or like partially dependent on proximity or contact to other cells of the inner retina, for example, like Mueller glia. I don't know if anyone's gone.
I don't think, I mean, what we find that, that some IPRGCs, the dopamine releasing amacrine cells synapse onto some of them. The synaptic stratification is different for these different subtypes, but I don't think anybody's looked at that detail. There are some mice where, so it turns out that there's also huge heterogeneity in transcription factors, and so with M1s there are two subtypes, and so if you knock out all the brain 3B positive cells, IPRGCs, there's a small subset of ganglion cells that are left. And they're enough, about 200 of them, to photoentrain and cause the pupillary light response. So, I mean, I think this system is probably the ancient system. A very few cells does a whole host of things. It's pretty amazing.
Thank you.
Hey, Phyllis, fascinating work. And just like you said, the complexity of those cells never ceases to amaze me, especially as somebody who thinks about rods and cones, which seem to be much simpler in regard. I have two questions. Very interesting that dopamine-dependent PKA modulates melanopsin signaling. So I guess the obvious question is, has anyone looked at the possible circadian modulation of melanopsin signaling?
Oh, yeah. Well, so these mice that we have don't have a huge circadian component because they're the melatonin's been knocked out. There is a light dependence on it. You're right. So I'm sure that there is a circadian component to dopamine in wild type mice. So I'm sure there's a circadian component to it.
But nobody has looked at that. It might be interesting in humans to actually look at the PLR and see whether there is a circadian regulation of that. And my second question is anything known about the calcium modulation in IPRGCs and whether calcium plays a role in adaptation of those cells?
Oh, that's a great question, and nobody's looked at it.
So there is still work to be done, huh?
Yeah, I'll keep you employed for a little bit.
Thank you very much for this wonderful presentation. I'm Narcisa from Dr. Kefalov lab. My question is about perception of contrast, that in every textbook and reviews about IPRGC cells, it is claimed to start in these cells.
In M4s.
Yes.
Which are alpha cells. For those who are retinal biologists, the M4s are traditional alpha cells.
Yes. In our retinal degeneration studies, we often do the testing of the visual acuity and contrast sensitivity with behavioral studies, and we want to correlate these results for a broader interpretation with objective measurement of the visual function with ERG and with morphology on the OCT or histology. So when we know that perception of fine details is in the rod and cones, so when we want to interpret visual acuity, we compare the results and correlate the results with A wave from the ERG that is given by the rod and cones photoreceptors, and with outer nuclear layer or photoreceptor layers on the OCT. But now my question, because we have this debate in the lab, do you think that contrast sensitivity, because the contrast perception is in the RGC photoreceptors, should be correlated with B wave of the ERG that is in the second and third neuron, and also the other layers of the OCT?
ERGs are beyond my expertise. I would say that there is a significant contribution of these cells, and so it might be interesting to create a mouse that is an RD mouse that's a melanopsin knockout, and look at your contrast sensitivity and see how it is different from your RD mouse with melanopsin. But I will bet you that there is a significant contribution of these IPRGCs.
Hi, Dr. Robinson. Very nice talk. I'm a postdoctoral scholar in Dr. Kern's lab, so I'm interested in retinal vasculature. And it is my understanding that activation of melanopsin causes a reflex, a vasodilation, a vasoconstriction of the vessels. So I was wondering if in your mouse model, have you looked at the retinal vasculature, what happens, in your knockout model? It's okay.
Because I know in the heart, there's OPN5 is also involved in the vascular constriction and dilation.
All right, we had a great conversation, not only the lecture, but the conversation as well. So thank you, Phyllis, and please join me to thank our speaker.